Okay. Good morning, everyone. Welcome to the session. Welcome to the session on the topic of molecular structure and types of immunoglobulins, or we can say it antibodies. So, in this lecture, our main aim will be first of all to clearly understand what is immunoglobulin, what is antibody, what is the difference between these two, what is the structure that commonly we are knowing as Y-shaped structure, what are the components of the structure, then what are the different fragments that we can attain after enzymatic treatment and how these immunoglobulins are responsible for providing the cell mediated immunity and what type of immunoglobulins are there, and in that type what is the basic structure, is there a common structure or there are multiple types of structure within a single type. So that would be our motto. We'll be starting from very basic part. Please listen carefully throughout the lecture. We'll be starting from very basic part and we'll be going till the end of understanding clearly. After this lecture, there should be a clear cut understanding what different types of antibodies are there in our body. In those antibodies, what are the different structures that we'll be talking about, and which one is the major antibody, which comes first, which comes in secondary responses. That notion will be settled down or laid foundation will be laid in this particular lecture, and in subsequent lectures we will be seeing that we'll be using these terminologies to build up the lectures and to have a clear understanding. So when we talk about immunoglobulins, I have just written two words here, Ig and Ab, to tell you clearly. Whenever you are reading anything, Ig means immunoglobulin and Ab means antibody. So why there are two terms? Two terms are there simply because immunoglobulins are the initial molecules that are formed by the plasma B cells. They are on the surface. The moment these immunoglobulins are released from the plasma B cell into the bloodstream and they're specific towards an antigen, those immunoglobulins are called as antibodies. So the difference is amongst the release of the cell towards the release of the globulins towards a specific antigen. Now, what is antigen? See, antigen is a foreign particle which is entering inside our body. Now, antigen should be a different character. It should be a foreign material. There are classes where we say that endogenous antigens are also there, but we'll see in later stages. For now, you should have a clear-cut understanding that a molecule which is foreign to our body, foreign means it is not a part of our body. It has been acquired from the environment. That particular molecule is called as antigen. And second important characteristic of that particular molecule is it should be able to evoke the immune response and bind to the immunoglobulin or antibody. That molecule is called as antigen. So by definition, if you talk about by definition, then the definition clearly says that the glycoprotein molecules that are produced by plasma B cells. So immunoglobulins are produced by a single cell type inside our body, and those cells are B cells. B cells. Among the B cells, there is a type called as plasma B cells. These plasma B cells are the active manufacturing unit of immunoglobulins or antibodies. So if a question comes, which cell type is presenting the antibody? It's very clear. There should be no confusion. It's the plasma B cells. It's the plasma B cells. Now, when these plasma B cell or the B cell will realize that it has to form an immunoglobulin only and only when an exposure is there for an immunogen or an antigen, which will result in the formation of antibody. So that is the reason why we are defining immunoglobulin as the glycoproteins which are formed from plasma B cells in response to an immunogen and which can function as antibody also. That is the definition of immunoglobulins. Then let's go ahead and look at this particular, yeah, it should clearly visible. Look at this particular graph that I want to showcase you. Now, where we are talking about the different components of the globulin, and the amount of protein that is present inside the serum, So when we say inside our blood, what is the percentage that we are talking about the immunoglobulins that is there? So the percentage is very high, and the percentage which is bound to antigen is low. is low. So we have a huge army. We have a huge army of this particular immunoglobulin, and amongst that army, the molecules are going, immunoglobulins are going and binding to the antigen, and that is their actually general function, that they should be able to bind to the antigen. By binding to the antigen, or the antibodies, when they bind to the antigen, they will result in the protection. The antigen will be cleared from the body. The mechanism will be discussing. And then they also bind to the antigen to provide a tag, so that the other cells that we have discussed, they can come and recognize the antigen where it is present and can help in clearance of that particular antigen from our body. And another part is the effector functions, which is in line to that particular antigen binding. The immunoglobulins will be binding to the antigen, and subsequently they will activate the other pathways. Let me just showcase you what I meant. Suppose we have an antigen here. The antibodies or the immunoglobulins have been released, so specific towards this. Now these are called as antibodies. Now, among them, a molecule has gone and bind to here, another molecule has binded here. So, what they have done, they have tagged this particular antigen. Now, the macrophagic cells will be coming, the complement proteins will be coming. They will never recognize this antigen, but they will recognize the antigen antibody complex. They will recognize this particular region, and in turn, they will be binding. They will be binding here. So what this antibody is doing, this antibody is actually providing the linkage or the anchorage for these complement proteins or the other cells which are required for the clearance of that pathogen. They will be utilized as tagging molecules. So that is the main function of these are the main functions of antibodies, that they go for effector function, and along with the effector functions, they also go for antigen binding and clearance. And if you talk to me that what is the end point is to clear the pathogen and to generate the immune response by activating the cells. That is the common function of immunoglobulins actually. So when we are talking about its structure, it should be very clear. This is the Y-shaped structure that we always talk about in books. You since beginning, everybody is making this kind of structure, right? This is the structure that people make, and this is how people are also responding. But in actual form, this is not linear. This is this is formed by these loop-like domains. Now, what are these loop-like domains? These domains are called as immunoglobulin superfamily domains. So I also told you earlier also that in whatever proteins, if these loop-like structures are present, then they will be clubbed under a single family called as immunoglobulin. That is the characteristic feature of this. These loops are characteristic features of immunoglobulin superfamily. Now, let's go ahead. Well, how this particular antibody or general structure of antibody is made. See, there are two chains. There is this particular chain which is continuing. Fine. This is called as heavy chain. And we have a light chain here also, which is partial. So we have two identical copies of light chain, one and second. These are two identical copies of light chain, which are almost 25,000 Dalton each. The molecular weight is 25,000 Dalton each. And we have two copies of heavy chain. Fine. So whenever you are understanding the structure of immunoglobulin, please understand that we have two light chains and we have two heavy chains. It is clear here also, two light chains, two heavy chains, right? Okay. Then how these chains are associated with themselves? Then let us see how these chains are associated with themselves. These chains are associated with these bonds called as disulfide bonds. Now, what are disulfide bonds? The red color bonding pattern, which are shown in this particular image, is actually disulfide bonds, and a single antibody contains around about 14 disulfide bonds that we talk about. 14 disulfide bonds. The bonds are in this manner. Now, how these bonds are formed is very common. If you have understood the biochemistry, if you know the protein or amino acid interaction, there is cysteine residues. There are cysteine amino acids. So when a disulfide bond is to form, it is formed between two cysteine residues. Why two cysteine residues? Because in them there is a free thiol group. There is a free thiol group, and a bond is formed. And a bond is formed in this manner. In this manner. So this two thiol moieties will be forming a bond, and this bond is called as disulfide. So if a molecule which is present here, another molecule of cysteine which is present here, they are forming a bond. So what will happen to the region which is actually present in between? It will loop out. See this. If a cysteine is here, another cysteine is here, and if they form a bond, what is happening to the remaining portion? It is looping out. So that is the reason why disulfide bonds are responsible for proper folding of the proteins. And apart from disulfide bonds, there are hydrogen bonds, there are non-covalent interactions that goes and that are stabilizing this particular structure. Apart from this, we always say that immunoglobulins are glycoproteins. Glycoproteins. So the proteins are conjugated with a carbohydrate. That's why we are calling it as glycoprotein. And the carbohydrate attaches always to the heavy chain. Always to the heavy chain. Fine. That is the reason why we are calling it as the why we call it as a glycoprotein. Glycoproteins are called as glycoprotein. So this is the structure that we are talking about. Now, among this structure, let us start with the nomenclature of this structure. We, apart from the chains, in the chains we have two different regions called as variable region and a constant region. Variable region and a constant region. Now, if I bifurcate this particular part in this section, this is variable region and this is constant region. So what we are how we are nomenclating. Let us talk about the heavy chain first. From where the heavy chain is starting? This is your heavy chain. Two identical copies of your heavy chain. So when I'm talking about the heavy chain, see, this is constant heavy region 1, second region and third region. So there are three constant regions in the heavy chain. There are three constant regions in the heavy chain and one variable region in the light chain. One variable region in heavy chain. So a heavy chain is containing total of four domains or four regions, out of which three are constant and one is variable. Now, when we are talking about your light chain, so we are talking about this particular portion and we have two identical copies of this portion. So in light chain, what we are having one constant region and one variable region. One constant region and one variable region. Thereby, the total regions are 2. So you see, this is how the bifurcation is there. Now, let me just remove all the ink and then let's talk about, if I say how many variable regions are present in an antibody, the answer will be 4. Why? These are the variable regions. 2 variable regions of each chain of light and 2 variable regions of each chain of heavy chain. And when you're talking about constant region, then we have these regions which are called as constant region. Now, in between them, there is a region which is represented here called as Heinz region, and it is responsible for providing the flexibility to the immunoglobulin molecule. So that is the thing that you should always remember, the Heinz region and how this particular structure is going about. Now let us see in much more simpler manner. Once we have understood the domain part, let us focus on this part, that how it is going and how the nomenclature is there. See, immunoglobulin is nothing but a protein. A protein is always synthesized from N terminal to C terminal. Similarly, this antibody always have N and C terminal. At the N terminal, there is a free NH3, and at C terminal, there will be COH, a carboxyl. So please be clear in your mind. The regions which are variable, they contain NH3 region. Regions which are constant, they contain a free carboxylic acid group. Fine. And that is the reason why the synthesis takes place from mRNA in this direction. Okay. Then Apart from this, let's talk about other scenarios. The variable region is presented here. This is the region which is responsible to bind for binding to an antigen. This region will be responsible for binding to the antigen, and this region, the constant region, is responsible for binding to the complement systems, or you can talk about the macrophages. We are talking about the macrophages, whatever the cell type we are talking about. The constant region is responsible for activating the other or recognizing the other immune cells, whereas antigen binding domain is actually the variable region. Is actually the variable region. That is why we are talking it as antigen binding site, and we also call this particular region as fragment of antigen binding. Fragment antigen binding. Fine. And this particular constant region is also called as fragment constant, which is one constant of light and three constant of heavy. So that is how the bifurcation is happening. And in between, the complete structure is stabilized by your disulfide bonds as well as hydrogen bonds, hydrophobic interactions, and multiple units. So moving ahead, let us focus on the same picture that we are very common and clearly established that these are domains that we have discussed. These are the domains that we have discussed. Now we have three constant and one heavy in case of heavy chain. But there are antibody types where we have also seen an additional constant region in heavy chain, but that is a specific cases. These are the specific cases that we are talking about, and not a general case. And of course, it's a glycoprotein that we are talking about. And let us focus on these slides. Will narrate. These are very common questions that do come in every competition. So, what we are trying to answer here, what we are trying to narrate in this particular section. See, if you treat the complete immunoglobulin with an enzyme called as papain, this papain will cleave the hinge region, at this place where you are seeing, and at this place there is no disulfide bond. So what it will be resulting in? It will result in separation of these regions. Now this single region is called as Fab, fragment of antibody binding. Fine. So there will be two Fab fragment and one fragment which is constant. So what is happening? Papain has cut cut the immunoglobulin. It has inculcated a cut inside the immunoglobulin in such a manner that it is releasing the two molecules of Fab and one molecule of fragment constant. This will be completely heavy chain. This will be light and heavy chain and light and heavy chain. That you should always remember. And what will be happening? This now contains single site of antigen binding. See, when we are talking about this structure here, so antigen can bind in this manner, antigen can bind in this manner, or in this manner also. So what we are saying? We are saying that we have two binding sites for antigen inside the antibody or immunoglobulin. Now that site is represented as valence, the valence of antibody. So it is currently 2 bivalent. So when we are talking about this particular Fab fragment, which is clear, now it has a single site which can bind to your antigen. This single site bind to the antigen, and that's why we are calling it as single valence. So you should remember what happens when you're talking about your papain part. Then when we are talking about this structure function relationship, see, this is what I meant to say when we are talking about the antigen binding. This is the site when we are saying that it can bind to the receptors which are present on the cell in order to activate the immune cells. This particular domain, the third domain of concentration, is actually responsible for transferring from mother to the fetus. It can cross the placental barrier based on this particular region. If you remove or mutate this particular region, the antibodies will never be able to cross the placenta. And the second domain of the constant region is actually responsible for activating the complement pathway, which is a part of your innate immune system. Now, next, enzyme that we should talk about is pepsin molecule. It's pepsin enzyme. Now, first we have talked about the pepin. Now we are talking about the enzyme called as pepsin. Enzyme called as pepsin. Now, what happens in this pepsin molecule? See, this pepsin molecule will cleave the antibody in the constant region. It will cleave this particular constant region completely. It will degrade it completely. So what will happen? You will never get the constant region in this scenario. The FC peptides is cleaved. So there are small fragments of FC, but an intact upper portion is retained with hinge region. If hinge region is there, then these arms of Y shape will never be separated. They will always be joined. And that is the reason why we are calling it as fragment of antibody binding dimer. We are not calling it as two molecules of Fab. We are not calling it as two molecules of Fab. Now they are not single molecules, but they are two molecules which are clubbed together, which have come together because of the presence of Hinge region. And that is the reason why we are calling it as Fab2. So the question comes like that: if you treat the antibody or an immunoglobulin with pepsin, what fragments you will be getting, and options will be in front of you. So what you have to clearly say, you have to see the option where this particular orientation is there or nomenclature is there: fragment antibody, fragment of antibody dimer along with FC peptide, along with cleaved SFC peptide. And when we say that what will happen with the papain enzyme that we have discussed, then you can easily say that it will be two Fab fragments and single fragment crystallizable. So there is a clear-cut difference between the options that you will be seeing. There is clear-cut difference between the option that you will be seeing in that scenario. So with that, basic platform of understanding how immunoglobulins are looking, let us see how many different types of antibodies or immunoglobulins are there. So there are 5 different classes that we already know: G, A, M, D, E. For your sake, you should always remember in this manner. I always tell my students to remember in this manner: gamed. If you are gamed, that means you are talking about your immunoglobulin class, and each class is called as a single isotype. When you are reading a word isotype, that means the classes what we are talking about, and we'll discuss in detail what are isotype, allotype, and nucleotypes. Okay, so among these classes, let us talk when we are talking about the human immunoglobulin. We say that IgG and ADE are nomenclatured as based on the heavy chain. This is gamma. This M stands for mu, A is for alpha, D is for delta, E is for epsilon. So this is how we nomenclate them in terms of human immunoglobulins. So the basic nomenclature is same, so you should be able to remember. Now, among all the classes of immunoglobulins, there are 2 classes that actually have further subtypes. Now, IgG, when we are talking about immunoglobulin G, it contains 4 different subclasses. It contains 4 different subclasses: IG1, G2, 3, and 4. Why these classes are arising? Because of the heavy chain sequencing. When you see the sequences of the heavy chain, there are 4 different types of heavy chains in this particular category of IgG. That's why we are putting them as gamma 1, 2, 3, and 4. And there are 2 different categories of IgA. That's why we are putting it as alpha 1 and alpha 2. And this is the what you can say the classification of immunoglobulin that you should remember. We have 5 different types of immunoglobulins: G, A, M, E, D, gamma, alpha, mu, delta, epsilon. And then we have among those subclasses, we have subclasses of IgG which have 4 different types of IgG, and in IgA we have 2 different IgG. So when we are talking about the immunoglobulins, how we nomenclate them. So the common name is what I have clear. When we are talking about IgM, in many books you will see that we nomenclate as kappa, and when we are talking about IgA, when we call it as lambda 2 and IgG. So the nomenclature is based on different light heavy chains that we are talking about, and that is actually responsible for the characteristic that we are calling it as heterogeneity. So let us talk about IgG in detail. IgG is nothing, it's a monomer, it's not a polymer, it's a monomer which is of 7s unit. 7s when we're talking about a sedimentation rate or Sydberg unit. Okay, So when we say that it is of four types, among those four types, let us see. The three types have a very common structure. The three types G, 1, 2, and 4 are very common, similar in structure, but 3 has an extended hinge region. 3 has an extended hinge region with multiple disulfide bonds, and that is the reason why we have different categories. Now, among the properties, if we talk about this is the major immunoglobulin. IgG is the major immunoglobulin of your serum, which is responsible for providing the immunity across the body, systemic immunity, and it is a major immunoglobulin if you're talking about the spaces or the extravascular spaces that we are talking about. Now, in terms of placenta transfer, this particular IgG can cross the placenta barrier when we are talking about IgG2. Among the IgG4 is responsible for activating the complement system, and Ig2 and Ig4 both can activate phagocytosis. They can activate the natural killer cells. They can activate the antibody dependent cell cytotoxicity pathways. That all those pathways, of course, you'll be reading in detail, but you have to understand first that in serum the major immunoglobulin is IgG. If it's major, then it will be responsible for mounting a very handsome immune response. And among the extravascular spaces, this is the anti-immunoglobulin which is present at higher quantity. It can cross the placenta. It can activate the other immune cells also. Then, when we are talking about this IgG, it should be very clear. It should be very clear that amongst the body, it constitutes almost 70 to 80% of the total immunoglobulins. Of the total immunoglobulins, this is the major immunoglobulin that is present inside our body. Fine. And among if you talk about the rate of production, then on 24-hour basis, our body, if it is forming 100 molecules of immunoglobulin, that the maximum molecules of IgG will be formed by our body. And in the plasma half-life, in the life inside our body, it is having the longest half-life among all the types, that is of 23 days. The half-life that we are talking about is 23 days inside our body. Then these are certain kernel characters that we're talking about. It contains four classes that we all say. Why we are talking as four subclasses? Because in this, the shape is okay, but the amino acid sequence that is present in this constant region, that is the reason why we are having four different types of immunoglobulins, four different types of heavy chain at the constant regions. Now, these subclasses may vary in different biological functions because their sequence is different. As we saw it, like in Ig3, we had a very long hinge region. We had a very long hinge region with 11 disulfide bonds. Then it is the immunoglobulin that can cross the placenta and provides the immunity to the newborn or the fetal stage. And among all the subclasses, Ig2 has the poorest ability to cross the placenta, and Ig3 is one of the very effective immunoglobulin when we are talking about among all those types. So if you have an option that amongst Ig1, 2, 3, 4, which is the molecule that cannot cross placenta, you should always tick or pick your answer as Ig2. Now, when we are talking about complement fixing, the Ig3 is the best one, followed by 1 and 2. Whereas Ig4 will never take part in complement fixation. Whereas phagocytosis, all four are able to go for phagocytosis. So these three functions in this slide are giving you an opportunity to answer various multiple choice questions. Okay, so what is the answer? If talking about the placenta crossing, Ig2 cannot do that. If we are talking about the complement fixing, Ig3 is best and Ig4 will never be doing phagocytosis. All of them can be performing the same function. Now, apart from this, the antibodies, these immunoglobulins are also responsible for neutralizing. Now, how we are talking about precipitation and neutralizing? See, there is an antigen. Now, these antibodies are released. These are immunoglobulin IgG. These IgGs are released. And there are three molecules, suppose, and we have multiple copies of immunoglobulin. They will go and bind to the antigen and they will form this particular kind of structure. What they are doing, they are actually immobilizing They are actually immobilizing or precipitating the complete antigen. So this is what they have done. They have aggregated the soluble antigen, and this particular complex will be very thick and it will precipitate out. It will precipitate out. That is called as precipitation. Now, when we're talking about neutralization, suppose a virus, a bacterial pathogen is there, it wants to infect the T cell and its receptor is ready. It will be binding here. In before binding to its receptor, an antibody attaches here. Now, what has happened? It has blocked, this antibody has blocked the interaction with its receptor, thereby it has neutralized. Thereby it has neutralized. So it is used in multiple functions, as we understand. Then next type is IgM. Then next type is IgM. So when we are talking about the IgM, see, IgM has the highest molecular weight. Why? Because it's pentavalent. It is secreted in form of pentamer. Pentamer. Now let us see how we are talking about as a pentamer. See, this is the structure that we are talking about. Each immunoglobulin is represented by a single IgM. Right? Now, five different IgMs or mu chains are coming together associated with a protein called as J chain, and they give rise to a pentamer kind of structure which has five binding sites of antigen. Five binding sites of antigen. And that's why it is called as pentameric form. It is linked with J chain. And this is the actual secreted structure of IgM, and it looks in such a manner. The J chain is actually responsible for cross-linking the immunoglobulin M in immunoglobulin. Now, they exist in this form also, and they exist in monomeric form also. They exist in monomeric form also. So when, suppose this is plasma B cell, so when IgM is expressed on their membrane, they are monomeric. They are monomeric. The moment they are secreted from the cell outside, they form this type of structure. So always remember, when the immunoglobulin M is membrane bound antibody as B cell, it is always monomeric. But when it is secreted, it is secreted in the form of pentamer. It is secreted in the form of pentamer. What are the functions? It has a very acute function. It will go and neutralize the antibody. It will not be having a very high half-life. It will act as antigen receptor. It will help in precipitation and agglutination, as we have discussed. And it is the first first effector immunoglobulin, which is released from the B cell in response to the antigen. And among the concentration, it's the third highest in the serum content, and it's the first immunoglobulin that is prepared by the fetus and the B cells. B cells first of all prepare your IgM, and the fetus also first of all prepares your IgM, and that is the antibody or the immunoglobulin that is secreted first of all in response to any antigen, and it can also lead to the activation of complement part. Apart from that, this antibody, because it's pentavalent, it can aggregate effectively. It can bind to multiple antigens and can form a cluster also. And it is used. It is used as a marker to identify plasma B cells also, because the B cells will always be having a IgM and IgM on their surface. Will be having always be having IgM on their surface, and that is used in identifying this particular immunoglobulin. Now I told you that we also have a case where we have a constant region 4, constant region of heavy chain 4. So let us talk about this particular slide. We have variable region, constant region 1, constant 2, constant 3, and we have an extra constant 4 in case of IgM with the extension of tail piece, and here there will be a free carboxylic acid. So this is the case that we were talking about, that in a class of IgM, it also contains this particular scenario. So I hope till now the message is pretty clear what we are talking about, in what terms we are talking about, and in what scenario we are discussing this particular aspect. So what we have discussed till now is nothing but it's a mere understanding of how to proceed with the things. So we have discussed IgA and IgM and basic structure completely. So let us talk about certain certain MCQs. These MCQs will be helpful for you to brush up what we have understood, like which is the most abundant immunoglobulin, how IgG is existing. So these MCQs and other MCQs also that will be coming in subsequent lectures will be helping you to understand basically how these events are important and which segment is to focus more on. So that is all in today's lecture. Thank you so much for your patience listening and wish you a very, very happy learning. We'll again meet with another lecture in the same series. Till then, stay safe and thank you so much.